Alright, so today we'll talk about the regulation of glycolysis. Regulation of glycolysis. I already made you to understand that there are three main steps in glycolytic pathway. Only three steps are important to us, just three of them. And what are those three steps? We have step 1, which is catalyzed by exokinase or glucokinase. Is that also? Then we have another step, which is the third step, I think so, catalyzed by phosphofructokinase 1. Is that also? Then I think the last step, which is catalyzed by pyruvate kinase. These are the irreversible steps of glycolytic pathway. Exokinase or glucokinase, phosphofructokinase 1, and pyruvate kinase. These are the irreversible steps of what? Of glycolytic pathway. Alright, so we move further. Please don't mind the noise. We'll proceed. Now, for you to understand the regulation of glycolytic pathway, I will just give you guys a table. In that table, on one part, I will have enzymes. On the other part, I will have their activator. And on this side, I will have the inhibitor. Okay? Alright. Now, I will take the first enzyme, exokinase. Okay? Exokinase is found in all cells. And what do you think will be the activator of exokinase? Now, exokinase, it doesn't really have a well-defined activator, so we can just put glucose there because glucose is the substrate. So in the presence of glucose, we expect exokinase to function better. But for the inhibitor, it is well defined. That is the product of that step, which is glucose 6-phosphate. Okay? Glucose 6-phosphate will act as a feedback inhibitor for exokinase. And the next enzyme is glucokinase. Glucokinase again is not really subjected to activation or inhibition by substrate. Rather, it is more or less induced. It's an inducible enzyme. What am I trying to tell you? It's not always present in the cell. It only gets induced in the presence of high levels of glucose. So basically, insulin will activate it. So it is under hormonal regulation. So insulin will act as an activator for this enzyme. How? It is going to induce the production of the enzyme. Let's go over it again. There are three steps of glycolytic pathway that are very, very important: exokinase or glucokinase, PFK1, and pyruvate kinase. Please don't forget that pyruvate kinase is also an enzyme responsible for what? Substrate level phosphorylation. I just said I should say that. Alright, so glucokinase, I said it is not always present in the cell. It only gets induced by what? Insulin. So insulin will act as the what? As an activator. Then moving further, let's talk about the third enzyme there, which is PFK1, phosphofructokinase 1. Are you there with me? Phosphofructokinase 1. Now, this enzyme is a rate limiting enzyme in glycolytic pathway. Let me just put it here: rate limiting step or rate limiting enzyme of the glycolytic pathway. Now, by rate limiting step, we mean that it is the most important step in the pathway. Without this step, this step is what determines what goes on in that pathway. If you can inhibit this step, you inhibit the entire pathway. Are you there with me? Alright. Now, so what do you think will activate this enzyme, and what do you think will inhibit this enzyme? Of course, when you have a high amount of ATP, which is what you want from glycolysis, then also, what is the aim of glycolysis? Do you remember? To produce what? ATP. Then also, It means that when the ATP level is high, will you want to do glycolysis again? No. So what do you think what do you think we inhibit this enzyme? I've already mentioned it, which is what? ATP. Anything that tells us that the cell is energy rich, we act as a what? As an inhibitor of the enzyme. Do you know another thing that can give us a signal that this cell is energy rich? I believe you are thinking citrate, because citrate is a sign that that cell is energy rich. Remember, okay, we'll get there. When we want to do what we call fatty acid biosynthesis, one of the first things to get from the mitochondria is what we call citrate. And the only time you can actually synthesize fatty acid is when you have a lot of ATP. So that's why citrate is a sign that the cell is energy rich. That's why citrate will inhibit PFK1. Are you there with me? Of course, nobody need to explain to you that ATP will inhibit it, because ATP is obviously a sign of energy. Is that not so? So ATP will inhibit it. Now, what do you think will activate the enzyme on the other hand? I believe you will be talking about the opposite of ATP. Is that not so? So you tell me things like what? AMP. Is that not so? AMP will what? Will activate the enzyme. But apart from AMP, there's one powerful activator of this PFK1 enzyme. Hello, are you there? There's one powerful activator of this PFK1 enzyme that is called what? Fructose 2,6 bisphosphate. Please, even if you forget ATP, even if you forget AMP as an activator, please don't forget this enzyme. It's very, very, very important. Fructose 2,6 bisphosphate, very, very important. I can't overstress that. Please, fructose 2,6 bisphosphate is a activator, it's a more powerful activator of PFK1 than even AMP. Okay, so I will explain more on fructose 2,6 bisphosphate as we proceed in this lecture. Let's go to the third enzyme. I will come back to that story about fructose 2,6 bisphosphate. So the last enzyme there is called pyruvate kinase. Pyruvate kinase. So pyruvate kinase, again, this is also an example of a rate limiting substrate level phosphorylation enzyme. Is that also? So this pyruvate kinase enzyme, the important thing about it is that it is inhibited also by ATP. It is inhibited also by ATP, which is high levels of energy. Is that also? And again, it is also inhibited by alanine. We will get to know about this in the protein metabolism lecture, where I will explain to you guys that, in fact, I will soon talk about it when we talk about the fate of pyruvate. You will now understand that pyruvate can actually form alanine, and this alanine can, in a way, act as an inhibitor of this pyruvate kinase. Because the only time you will think of, say, you want to form protein from carbohydrate is when you are actually energy rich. Are you there with me? So alanine can also inhibit what? Pyruvate kinase. For simplicity's sake, just remember that ATP is an inhibitor of pyruvate kinase. Also, alanine is an inhibitor of pyruvate kinase. Then what do you think would be the activator? The activator is fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. You know that is the product we got from PFK1. Fructose 1,6 bisphosphate. Are you there with me? So this one will act as a feed forward mechanism. Feed forward. It will act as a feed forward mechanism, meaning that it will go down the pathway to open the way more. So when you are forming fructose 1,6 bisphosphate, some of it will go to pyruvate kinase enzyme and activate it before the glycolytic pathway will get to that point. So it's called a feed forward mechanism. Are you there with me? All right. So in this table now, I've been able to explain the regulation of glycolytic pathway, starting from exokinase, glucokinase, PFK1, and pyruvate kinase. I made you guys understand that glucose 6-phosphate will act as a feedback inhibitor of exokinase. Then I just included glucose as an activator there. Are you there with me? Then I made you guys also understand that glucokinase is not under more of substrate activation or inhibition. Rather, it is what induced. That's the main word. It is induced by what? By insulin. So insulin is an activator of the enzyme. Then PFK1, which is the rate limiting step of glycolytic pathway. I made you guys understand that the main aim of glycolysis is to form ATP. So once you've gotten your ATP, you will not want to do glycolytic pathway anymore. Hence the reason why ATP and citrate act as inhibitor for PFK1. So what then is the activator? The activator is both AMP, which is sign that the cell is energy poor, and alongside fructose 2,6-bisphosphate, which I said is a more powerful activator of the enzyme. I will come back to that, how we actually arrive at fructose 2,6-bisphosphate. Then lastly, I made you guys understand also that pyruvate kinase is also an irreversible enzyme, and it is inhibited by ATP alongside by alanine, but activated by a what? But activated by a feed-forward mechanism from what? Fructose 1,6-bisphosphate. Is that also? Alright, so in the next video, I will explain on the mechanism behind the formation of fructose 2,6-bisphosphate and the fate of pyruvate.